Welcome, everyone. Thank you for joining us for today's webinar, Cannabis-Based Therapeutics, Indication, Formulation, and Route of Administration. It is presented by Guillermo Moreno-Sanz, PhD, founder and CSO of Abagune Research. My name is Judy O'Rourke, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Moreno-Sanz. I will now turn the presentation over to him.
Thank you, Judy, for the introduction. Thank you, everybody. It's a pleasure for me to give here this presentation of LabRoots. And what I'm going to talk about today is about therapeutic products that are based on cannabis or cannabinoids that have been approved by a regulatory agency, either the FDA or the EMA, the European Medicine Agency. And then we'll talk a little bit, or we'll investigate a little bit about the indications that are either approved or recommended for cannabis and cannabinoids, different formulations that we can use or we can find in the market to exploit this pharmaceutical action of cannabis or cannabinoids, and then how different routes of administration might be preferred depending on the indication and the formulation of cannabis and cannabinoids that we want to use. So basically what I'm going to do, it's a brief introduction to the therapeutic use of cannabis. We'll see examples of pharmaceutical drugs that have been approved by regulatory agencies, what clinical indications exist nowadays for cannabis and cannabinoids, and the routes of administration, as I was mentioning. Briefly introducing my company, it's called Abagune Research. We are located in the north of Spain, in the Basque Country, and it's my scientific initiative after years of working in academia, in particular at the University of California in Irvine. What we realize is that companies operating in the US are having difficulties working with their local universities because they operate under state law, while universities generally federally funded. So what Abagune brings to the table is the opportunity for industry leaders to have scientific consultation, also externalize their research services, and we specialize also on product development. So basically, cannabis and cannabinoids. Cannabinoids are a family of molecules that exist on the plant Cannabis sativa and are exclusive to this plant. Normally, the cannabinoids are synthesized in the flower or in the trichomes of the flowers and bracts. And the cannabinoids have one molecular precursor called cannabigerolic acid that can be converted into THCA, or tetrahydrocannabinolic acid, or CBDA, cannabidiolic acid, that upon heating by non-enzymatic decarboxylation, like in the oven when you bake them or when you smoke them, are converted to the neutral forms that we all know: THC, delta-9 tetrahydrocannabinol, or cannabidiol. The main cannabinoids, tetrahydrocannabinol and cannabidiol, are depicted in this slide. THC, it's a partial agonist of type 1 and type 2 cannabinoid receptors, or CB1 and CB2, as they are also known. The activation of central CB1, the CB1 receptors that we have in our brains, is responsible for the euphoric effects of THC, or what is commonly known as the high of THC. and has several reported effects as an analgesic, anti-inflammatory, appetite stimulant, sedating, antiemetic, and antitumoral, among others. CBD has been described as an allosteric modulator for CB1 and CB2. That's the reason why, while CBD it's psychoactive because it has some reported effects such as anxiolytic or antipsychotic, which are exerted by the action of CBD in the brain, it is not overly euphorisant, so it won't get you high. Other pharmaceutical actions for CBD that have been described are as an inhibitor for Thc activity, which is responsible for the degradation of the endocannabinoid anandamide, or also as an agonist for the receptors 5-HT1A, serotonin receptors, and vanillin receptors TRPV1. It also inhibits adenosine deactivation. So here we can see the reported effects for CBD, the most well-known or the best known as an anticonvulsive agent, recently approved by FDA in the presentation of Epidiolex for Dravet syndrome and Lennox-Gastaut pediatric epilepsy. Basically, the target of THC and the endocannabinoids, which are naturally occurring molecules in our body and our brains. The target for these molecules is called the endocannabinoid system, and is composed by some of the players I already mentioned, such as the cannabinoid receptors, CB1 and CB2, and the ends and the ligands that activate them, anandamide and 2-AG, and the enzymes responsible for the synthesis and degradation of these ligands. Basically, as part of my PhD dissertation, this is work that has been done at the University of California in Irvine and published in 2010 in Nature of Neuroscience. We discover or characterize how activation of peripheral CB1 receptors, that is cannabinoid receptors that are outside the brain, was sufficient to elicit cannabinoid-mediated analgesia, meaning that for pain management, or at least some forms of pain management, we could technically activate only cannabinoid receptors outside of our brain. We will have equal efficacy as what we do generally. I want to introduce a couple of concepts, basic pharmacokinetic parameters that will be useful for the rest of my talk. So basically, when we're talking about a drug that we intake, the typical pharmacokinetic parameters that we need to we care about is how this drug will increase its concentration in our blood. Right, we see how it goes up until it reaches Cmax, which is the maximum concentration that after a single intake we will have in our blood of this particular drug. And then we have a phase, this is a phase of absorption, and then we have a phase of elimination in which the drug concentration in the blood decreases. In between, there's a range that we call the therapeutic range, meaning that it's composed by the minimum concentration of the drug in the blood that we need to actually feel an effect or to have an effect. And then the minimum concentration that starts to be toxic, or in the case of cannabis or THCA in particular, starts to be discomforting, causing overly psychoactivity, or even some other adverse effects, such as dizziness or somnolence. And this will be the therapeutic range, the range of concentration in the blood in which we have efficacy and we have therapeutic gain without any side effects. The Tmax is the time that takes for the drug to reach the maximum concentration in the blood, and the time to onset means how long does it take for the drug to reach minimum effective concentration. So how long does it last between we intake the drug and we feel the first effects. So traditionally, cannabis has been normally smoked. Even NIDA used to distribute joints to patients. The viability of THC depends on the way of smoking. If you will take long draws or shorter draw, and how I don't know other particulars of your smoking device, and it's considered a very rapid route of administration. The time for the maximum concentration of THC in the blood is 9 minutes, And this allows the experienced user to better titrate the dosing by taking more or less hits. And the immediacy of the effect allows you to understand if you need another dosing or not. The problem with the smoking is that, as you can see here, the concentration in the blood goes over 100 nanograms per ml really fast. So 100 nanograms per ml, it's more or less what is considered the concentration of THC in the blood, after which you will have psychoactivity or you will be high. However, this is the preferred route of administration for many users, especially experienced ones that can, depending on the way of smoking, control the amount of THC that gets to their bloodstream, and therefore the efficacy of the treatment. So very briefly, pharmaceutical drugs based on cannabis and cannabinoids that have been approved by different regulatory agencies. We have Marinol, which is dronabinol, which is a synthetic form of THC. Now, this molecule is exactly the same as the THC that you will have in the plant, but it has been synthesized in a lab. And that allows it not to be a Schedule I drug license, but a Schedule III drug, a Schedule III substance, and therefore its use is much less restricted by the DEA. So Marinol come in a presentation of soft gel caps containing 2.55 or 10 milligram of THC. Excipients are pretty common: gelatin, glycerin, sesame oil. And it presents, and this is why I want to point out these characteristics about approved drugs. It presents high inter-individual variability, high absorption, but high liver metabolism. So 90 to 95 is absorbed, but only 10 to 20% gets the bloodstream. And high apparent volume distribution with high plasmatic protein binding. What does this mean is that THC doesn't like to be in the water. It likes to be binding either to protein or to fatty tissue, such as the brain or your adipose tissue. Another formulation of dronabinol, the same synthetic THC, is Syndros. This was approved in 2016 by the FDA for the indications of anorexia and VIH wasting syndrome and chemotherapy-related nausea and vomiting. So it has a liquid presentation instead of a soft gel cap. It reduces inter-individual variability, but it still has ethanol and propylene glycol. There's nothing fancy about the excipients of this formulation also. But it reduces the inter-individual variability, which is an advantage over Marinol. Thesamet is it's an analog. This is a synthetic molecule that is different in its structure from THC. But basically, the way of administration is the same. It's ingested as a hard cap, and the results are more or less the same that for THC. The only thing is it's a synthetic compound. It's approved also for chemotherapy-related nausea and vomiting, and it's a Schedule II controlled substance instead of a Schedule III or Schedule I in case of natural THC. Still, adverse effects are somnolence, dizziness, and euphoria, which are common to cannabis and cannabinoids. Now we get to a different set of approved compounds or approved pharmaceuticals, which are based actually on cannabinoids that come from the plant. They're not synthetic. The first one will be Sativex, which is a sublingual spray that contains 2.7 milligrams of THC and 2.5 milligrams of CBD per spray. Excipients, again, ethanol, propylene glycol, and an oil to give it a flavor. And it's approved only for spasticity associated to multiple sclerosis, although there are some clinical services that are using them off indication for, in particular, chronic and neuropathic pain. The difference is that it allows you to control dosing much better, and it's absorbed through the oral mucosa, at least partially, so it has a more favorable pharmacokinetic profile. It's absorbed faster than if you will take it only through your stomach. Finally, Epidiolex is the medicine that we mentioned before, has been approved by FDA for pediatric pediatric seizures in the Dravet syndrome and Lennox-Gastaut. Again, it's a transparent, colorless oral CBD solution, very, very concentrated. The weekly dose, it has a period of weekly dose escalation, the same that we see with all cannabinoid medication. It comes from 5, 10 to 20 milligrams kilograms per day. And because it's a very, very high concentration, it's a high dose of CBD, it has some adverse effects such as liver damage, somnolence, or suicidal thoughts. So how does it change? How does the formulation change? How is it different to take Marinol or Syndros when it's the same synthetic THC? Well, you here see the difference. When you take a capsule that has to go through your stomach and get absorbed into your intestine, it's not the same as taking something that is already liquid, that is going to be much more favorably absorbed. And here you see that after 15 minutes, in all these volunteers or on all these patients that took both formulations, 100% of those who took Syndros had already THC on their blood, and less than 25% of those who take Marinol had already THC in their blood, meaning that Syndros is going to have a much faster absorption than Marinol, and that's very important, especially for acute indications such as acute pain or epilepsy, in which you need a fast response. Also, the intra-individual variability is reduced in the case of Syndros compared to Marinol. You see here 13.5 versus 36 is almost half. So another example for changing the route of administration would be smoking versus a product like Sativex. So you can hardly see the numbers, but basically when you smoke, we talk about those 9 minutes that THC will take to get into your blood. We have data from THC extract that has been vaporized with a volcano, for example. The Cmax is going up over 100%, so you're going to have psychoactive effects coming from that THC. And the time for maximum concentration, it goes to 17 minutes. Instead, with Sativex, you have a much more gradient increase on the concentration. It stays around 5 to 10 nanograms per ml, and the Tmax is 60 minutes. It's a much more slower way of absorb your THC. Other formulations like a CBD self-emulsion that was published by the Atzmon group in 2017, compared to Sativex itself, you can see how the green line is the self-emulsion, the red line is Sativex. You see how you get higher concentration at a faster time, and you have linearity with the dose. In the figure below, you have a dark green, which is 100 milligrams, and you have a light green, which is 10 milligrams, is the same one that you have on the top. So this is just to give an idea how different formulations are going to affect the absorption of that CBD, which might actually cause a difference in efficacy. This is pharmacokinetic data from Epidiolex, and you can see here in the table how plasma concentration after one single dose reached similar concentrations of CBD. There was no difference between 5 milligrams kilogram per day, 10 milligram, and 20 milligram, probably because you have reached the saturation level of CBD, meaning that you cannot get higher CBD on your blood. Well, because it's being metabolized. You can see the metabolite 6-hydroxy and 7-carboxy CBD, how they go up as you increase the dosing, but the active compound in the blood is not going to be higher. So speaking of this type of clinical trials, actually this elimination phase, I mean, we all focus on how fast cannabinoids are absorbed, but how these cannabinoids are being eliminated or are they building up in our system and in our blood, it's also an important fact that many of these clinical designs are ignoring when we consider the last sample is normally taken at 72 hours, meaning 3 days after intake. So speaking, I mean, I just wanted to touch base really briefly on clinical indications for cannabis and cannabinoids, because this is something that we hear a lot, and we get asked, I mean, both clinicians and researchers alike, get asked by patients, right? Even if in our families, I mean, what is cannabis? I mean, will cannabis help me? So there's a lot of pharmaceutical promise on cannabis and cannabinoids, and I say cannabis because, I mean, it might be, we have high THC strains, we have low THC strains, high CBD strains. So composition of cannabis is never heterogeneous. It's never homogeneous, sorry, it's actually heterogeneous. But what we have basically agree, and it's collected in this excellent report from the National Academy of Sciences, published in 2017, that I have the honor to contribute as a consultant to, have found that we have conclusive evidence of the clinical efficacy of cannabis and cannabinoids for chronic pain in adults. 65% of patients that take cannabis for therapeutical reasons do so for pain indications, normally chronic pain indications, and also for the ability to reduce the use of opiates, prescription opiates that they're normally taking for the chronic conditions. It's also, there's conclusive evidence for the effects of cannabis as antiemetics for the treatment of chemotherapy-induced nausea and vomiting, and also for improving patient-reported multiple sclerosis and spasticity. So after this, I list another series of indications that the report describes as moderate evidence or no so conclusive evidence, and then we'll see such as improving short-term sleep outcomes in individuals with chronic conditions like sleep apnea or fibromyalgia, chronic pain, and multiple sclerosis. So these are other indications that we have limited evidence or clinical efficacy, like increasing appetite on weight loss associated to AIDS, and improving clinical measure multiple sclerosis, anxiety disorders. Other indications that we have anecdotic evidence. Patients are feeling relief on these indications, but there is no scientific data to support these indications for cannabis. And then we have limited evidence of a statistical association with better outcomes on traumatic brain injury, and same for dementia, glaucoma, or depressive symptoms in chronic pain and multiple sclerosis. I really invite the audience to revise at the least the outcomes of the report, because it's very, very interesting. And also, it brings some light about a lot of conditions for which there is this therapeutical promise of cannabis and cannabinoids, but unfortunately, we don't have enough data, clinical data yet, to support it. I'm a preclinical researcher, so I've been working with preclinical models most of my life. This means that I work with rats and mice. And we have seen cannabinoids working in animal models for many, many different indications. But this is the same case for many different drugs that are on development. How these drugs will behave once we take them to the clinic, it's a mystery to us. So although there's promise in animal models, we cannot simply conclude that we're going to see the same efficacy in humans. So basically, what I wanted to is to set the frame from what indications we can actually discuss about taking cannabis for. When patients with chronic pain or with nausea induced by chemotherapy or spasticity associated to multiple sclerosis, we know that we have data to justify the use of cannabis and cannabinoids. And, doctors and clinicians are going to feel more comfortable recommending cannabis for those. So what I wanted to see or to discuss is what approach it makes more sense depending on the indication for the intake of cannabinoids. So this is a summary of the different ways in which we can put drugs in our organism. We have the gastrointestinal route, meaning through the mouth, either by injection or sublingual through the oral mucosa. We have the enteral route for the liberation, the release of compounds in the intestine, and we have the rectal dose with suppositories normally. Parenteral route means IV, when we inject something directly into a vein. The pulmonary route is when we inhale something, and the topical route means through our skin or mucosas. So basically, we have systemic distribution. meaning that cannabinoids or any drug that we're taking will get to our blood through the buccal, sublingual, oral, transdermal, or pulmonary route. And these are potential therapeutic indications that will benefit from a systemic distribution of cannabis and cannabinoids. And the type of product that we could think of when we want a sublingual or buccal could be a mucoadhesive, like putting cannabinoids on a patch that you can attach to your oral mucosa. A tincture, such as Sativex, something that you got, a tincture normally in ethanol that you can absorb through your oral mucosa, or a troche, sublingual tablet, or a buccal tablet. When we think about oral route, we think about caps, syrups, oils, or chewing gum, so we can ingest cannabinoids. And then on the pulmonary, we have inhalation powders and mist, and also we have plant smoking or vaporizing. So some strategies to standardize the pulmonary route are, this is a titanium pipe that has been used in clinical studies to determine absorption of tobacco. So there's a company in Canada that has developed these pellets. So you have a more standardized way of smoking, which is surprising to me, because at the end of the day, probably the only agreement that the clinical community has on cannabis and cannabinoids is that you should not smoke. Smoke comes with with other problems to your health, especially for patients that have already, which health is compromised. And it's a way of administration that allows less control over the experience, although, as we saw before, experienced users might actually control the levels of THC in the blood by using this route. Other options are, of course, vaporization, either of herbal cannabis, such as the examples that I'm showing here, or vaporizing extracts, which is a practice that has been extended by the recreational market, but that some patients actually benefit from. And the other option will be by pulverization of either powder or a mist containing cannabinoids, which is a challenge given the lipophilic nature of cannabinoids, but still there are groups trying to develop products in this line. Sublingual or buccal products such as Sativex have a faster absorption through the oral mucosa, and have much less first pass metabolism, that they don't get degraded in the liver so much. A couple of examples are Eco Pharmaceuticals in the Netherlands, and a California-based company called Troche that develops these tablets. You can read more about this formulation on the paper we published in Frontiers of Pharmacology last year in August. Another route of administration that has been very popular because it has been developed by a company in the US is Transdermal Administration of Cbd. This image, it comes from a scientific paper published in 2010, in which this academic group tried to come up with a formulation that will allow the systemic administration of CBD, meaning CBD get into your blood and then distribute throughout your body through the skin. And using a series of excipients, they succeeded at getting CBD on the blood of these animals. I don't recall if there were guinea pigs, hairless guinea pigs. The benefits of having transdermic CBD absorption will be to reduce the incidence of gastroduodenal issues, and also reduce hepatic first pass metabolism, drug-drug interaction in the liver, and also avoid the conversion of CBD to THC in the stomach, which is still controversial. I think right now there's like 3 or 4 groups that have described the conversion in the acidic environment of the stomach from CBD to THC. That would be technically something that one could avoid with this route of administration. However, I think this conversion in vivo remains still controversial. This academic discovery has been developed by the pharmaceutical company Synerva, which has already 2 products in development, even in preclinical trials and clinical trials with different outcome, different success. Other routes of administration that are not necessarily systemic but more local could be rectal, vaginal, intranasal, and ophthalmic. So all these mucosas will allow for THC and CBD to get to the blood, but will also have a local effect. So rectally, we could think of inflammatory bowel diseases, patients that might have pain or inflammation located in a particular organ or area of the body. In this case, the colon could benefit from suppositories or enemas or gels rather than by ingesting something by mouth. Same goes with dysmenorrhea, endometriosis, other diseases from the endometrium. In women, intranasal, of course, will be a route of administration that allows CBD or THC to get to the blood, but also could be used for nasal diseases such as rhinitis or neurological affections, migraine, always with a local focalized distribution, and also ophthalmic through the cornea, the iris mucosa. Drugs can also get to the blood, but also will be aligned with some potential indications for cannabinoids, such as reducing the intraocular pressure in glaucoma. The formulation in this case could be eye drops, for example. So these are some examples from the recreational market that gives us an idea of what either vaginal ovuli or suppository containing CBD or THC will look like, and there's actually patients looking after this presentation and using them for some of these indications. What about intranasal administration of CBD? The same group that did the transdermal brought us some data about the absorption of THC and CBD through this route of administration. And this is an example. I think this company comes from Colorado. And, how we can successfully administer THC or CBD depending on the parameters that we want to play with. In this case, intranasal administration will be a fast way of absorbing cannabinoids that for indication that maybe cannot wait 1-2 hours for an onset of action, and then at the same time won't be preferred for chronic treatments in which we need to maintain the concentration in the blood within the therapeutic range. The more innovative the more innovation that we're seeing in the area of cannabinoid pharmacology, comes with the development of nano carriers for cannabinoids. Now, for cannabinoids and other natural compounds, this is a very nice review by Conte and colleagues, in which they revise what kind of techniques we have to make little vehicles for all these active molecules to get to the places where they have to go. In general, these molecules, especially in the case of phytocannabinoids, they're very difficult to dissolve in water, and therefore they're not very friendly, or they don't match with biological environments, because at the end of the day, we're 70% water. So there are many things that we cannot do on phytocannabinoids as we know them. For example, we know that oil and water do not mix, so it's hard to think about a water-based formulation for these guys. However, there are already companies and groups that are trying to challenge the solubility of cannabinoids in the water through some of these techniques. This could allow for for a range of applications for cannabinoids, because we've heard in the news about why not making beer, right, with cannabis for the recreational market. But at the end of the day, if we can put THC and CBD in an IV administration, we can put them in a syringe that we can inject intradermally or intra-articularly, then we will reduce the side effects that are associated to cannabinoids, because we will confine them in a particular space within the body. And we will maximize the efficacy and bring down the adverse effects. And of course, at the end of the day, why not? We will be able to enjoy cannabis flavored soda if that's what appeals to you. So basically, with these structures, there has been some academic work. There has been also some industry work that for which data is not really available. My impression is that there's a lot of CBD water out there that has no CBD at all, but that is my understanding so far. So the message I'm trying to send is that this is technically feasible. We have done it for other things that do not dissolve in water that are not cannabinoids. So cannabinoids wouldn't be, they're challenging, but we could find a solution for this. But definitely there's a level of technological refinement that is required to do so. So basically, the strategies that we have followed is making nanoparticles, either from polymers or from other lipids, in which we can load cannabinoids. And we have seen how, formulated in this way, cannabinoids increase their efficacy. At the end of the day, it remains a challenge to see if we can reduce side effects, but it seems that it will allow a control over the release of these cannabinoids, both in space and time, meaning that we can have controlled release formulation. So you don't need a daily intake, but a weekly intake, for example, of cannabinoids, or a site control formulations, meaning that even if you ingest this type of formulation of cannabinoid, it will be released only in the place where it is required, thus reducing the amount of side effects that might have. Applications of this formulation could be topical for a cosmetic or cutaneous infection, such as dermatitis or eczema. And it will have on the presentation of a cream or a gel. We could have intradermic. There will be still a local distribution. We could do this by injected intradermally or by coming up with formulations that were able to penetrate the dermis, the epidermis into the dermis. And there is where the site of action for these molecules it's found in many places. That's where you have your hair follicles, that's where you have your nerve terminals, and that's a way in which we can technically, theoretically increase the efficacy of these compounds. Finally, the intra-articular administration. We know that cannabinoids have a lot of therapeutical promise for inflammation and joint diseases. So being able to place these molecules right in the place where they are needed will probably and likely increase their efficacy also. So this is the end of my presentation. I really wanted to send this message about this education about it. I think it's worth thinking that there are many options for patients and for recreational users too, other than smoking. And depending on the goal of our practice with cannabis, some of them will be preferred over others. What we are maybe missing is a little bit of education and knowledge for all the users to have a safer and more effective use of your cannabis and cannabinoids.
Thank you, Dr. Moreno-Sanz, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through June 28th, 2019. And as a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Bye.